Hi everyone, my name's Lewis. I am a professor at the University of Westminster, and the role that I have there is a professorship in science communication. So half of my job is writing books, doing science engagement, science outreach, that side of things. And the other 50% of my time is on the research, is on the astrobiology. So as an astrobiologist, I came originally from a biological background. I read biological sciences at Oxford, moved to University College London, to UCL for my PhD, MRes and PhD combined program in interdisciplinary science, which is when I got involved in astrobiology. Spent some time at University of Leicester, University of Kent, and then did a loop full circle through the Midlands of the UK and landed back in London at the University of Westminster, 10 yards down the road from UCL where I started out. And the focus of my research in astrobiology is on Mars and the possibility of there being ultra-hardy microbial life forms in the near subsurface. So talking about extremophile type life. So I work with samples that I've either collected myself or have got from collaborators from Martian analog sites on Earth. So deserts on Earth that are like the Martian surface in some important respects. So places like the Atacama Desert or the Antarctic dry valleys, looking at different mineral samples, different bits of the surface and mineralogy. And then importantly, trying to understand what signs of life, what biosignatures could be detected of extremophilic life in these Martian analog type samples. And so the sort of instrumentation that I use are things like Raman spectroscopy, infrared spectroscopy, reflectance spectroscopy. So the sort of techniques that we are now sending to Mars on board our Mars rovers, on board our space probes, to see what sort of signs of life we might be able to detect using those. And sort of another wing of what I do in astrobiology is focusing on the cosmic radiation. So if you go beyond the protective cocoon that the Earth provides us with a magnetic field and a thick atmosphere, you become exposed to some very nasty radiation in outer space, the cosmic rays. And this is important if Elon Musk or NASA or someone starts sending humans to Mars, you start exposing your body to the radiation of outer space. But it's also important for any native indigenous Martian life. If there is Martian bacteria on the surface of Mars, they also are unprotected from that cosmic radiation. So another important question is, how quickly does that radiation kill any cells that might be dormant for a period of time? And then how quickly does that radiation erase or rub out signs that life was once there? How quickly do different biosignatures become degraded and removed by the radiation? So the sort of stuff that I do in astrobiology is very interdisciplinary in the sense that it involves a lot of biology, my own background, with planetary science, a bit of physics, a bit of instrumentation and engineering, and also different sorts of study. So working in the lab with some extremophiles, or working with instruments on samples that have been brought back from around the world, or doing computer simulation, computer modeling on what the radiation on Mars is actually like, by using the sort of computer code that they use at CERN for the Large Hadron Collider. That's how we model cosmic radiation. So that's a nutshell of the sort of things in general terms that I get involved in the astrobiology research.
Thank you for sharing with us this. And now I have, now I have some general questions to ask you. Okay, the first would be, how could you define life with 3 words?
I'm not sure you can define life in 3 words, but I think there are conveniently 3 things which are important to life, that life must possess or must be able to must have these functions in order to be alive. And those are information and metabolism and membrane. You need to have those three fundamental properties in terms of genetics and information, and in terms of biochemistry and providing energy for yourself. And then also in terms of membrane, having some kind of wall that defines you as a cell that is separate from the outside world and your environment. And you need to have all three of those things in the same place in the same time in order to be alive. And therefore, there's something of a three-way chicken and the egg problem when we start trying to work out how life got started in the first place. What was the origin of life, and how do you get genetics and biochemistry and metabolism and a membrane all together at the same time? Because any subset of those three isn't a fully functioning cell. How do you get up to that level of complexity in the first place?
Okay, thank you. The next one is because you have said that you are a professor in science communication. So what would you say to someone that says, I hate maths and science? How would you convince them?
I would say you're entitled to your opinion. I would say saying that you hate math and science, I would think is a bit like saying I hate food or I hate sports. You know, I find it hard to understand that someone hates every single aspect of science and maths. Although I do have a lot of sympathy for people that are turned off STEM subjects, science and technology and engineering, math subjects at school, because sometimes I think maths and science is taught as a list of facts you need to remember and regurgitate an exam or label this diagram. Whereas science isn't about facts that you remember. Science is about how you find those things out in the first place. Science is incredibly creative and imaginative, and it's a great problem-solving enterprise in that sense. And of course, there's other aspects of science in terms of how it's presented and making it seem relevant to you in your everyday life, and not just about, you know, atoms and molecules and galaxies that are far, far away. We live in a scientific world. If you catch coronavirus, heaven forbid that you do, but if you catch coronavirus, it's the science will help you get better again. And I think understanding and appreciating that is a part of living, you know, living in our modern world today.
Yeah. Okay. Again, thank you. That's, I think, one of the aspects I see as well in people our age, saying that they hate math or physics. Maybe it's due to how they've been taught at school. Yeah. And now my last question could be, which song could you personally send to other planets as a sign?
So I clearly, for my research field in astrobiology, I have a lot of time for. I really enjoy David Bowie's song "Life on Mars," and invariably, whenever I invited on to radio show to an interview, or there's been some Mars science result and discovery, and I'm talking to a DJ about it, it's almost always "Life on Mars," David Bowie, that they choose as the song beforehand. So I've listened to that song a lot and come to love it.
Okay, thank you. Thank you very much. Maybe do I have time for another question on?
Yeah,
Would
sure.
you like to go on, Joaquin?
Yeah, go ahead. Yeah.
Okay. Which one do you think that will be the reaction to the discovery of extraterrestrial life? I mean, not for the scientific community, but the society reaction in general.
Yeah, I think it's a very good question. And if we say the discovery of extraterrestrial life, let's say finding bacteria on Mars or Europa somewhere, and not little green men bundling out of a shiny UFO that lands on the White House lawn. Because I think finding bacteria on Mars is what we're likely to discover, most likely to discover. And I would be really curious. I don't know the answer to that question. I suspect it's a discovery that will be genuinely profound, and I think it will be on the front page of every newspaper around the planet. But I wonder what people think about it like a month later or a year later. I think it's just one of those important discoveries that gets absorbed into, the collective subconscious. People understand that and then move on with their lives. And within a couple of days' time, the news will be about the Kardashians and what they've been up to in their lives again, and celebrity gossip and the sort of things that fill the newspapers anyway. Whereas I think it'll have deeper repercussions in the scientific community. What do we do after? What do we next after finding life on Mars? How do we bring samples of that back to the labs on Earth so we can come to understand that life, see what makes it tick, see how it works on a molecular level, and therefore see how that Martian life is similar to us, perhaps, but maybe in other ways that it is deeply alien, that it's just built in a fundamentally different way. Because I think that would be truly fascinating to understand, to see how biology can work in different ways.
Okay, thank you so much.
Okay, so now we would like to talk also more in depth about your research and more prepare science itself. So I prepare a bunch of questions regarding your investigation in radiation and in extreme environments. So could you explain us a little bit more about the role of an atmosphere in a planet, and also why and how it's important, and on the other hand, how much important does it has, up to what extent it is important?
Well, firstly, if the Earth had no atmosphere, there would be no life on Earth, full stop. It's that important. It's 100% important, at least for life on the surface and not life a kilometer deep in the crust of the Earth. But the atmosphere is useful for a number of different things. Having atmospheric pressure enables water in the liquid state on the surface, enables oceans and rivers and lakes. The greenhouse effect of carbon dioxide and other greenhouse gases in the atmosphere blanket the Earth, but they keep us warm enough for liquid water on the surface. They trap some of the sun's heat. And the atmosphere is protective in other ways as well. Having a very thin layer of ozone in the atmosphere lets through the visible wavelengths of sunlight. So we can do photosynthesis, so plants can grow by the energy of sunlight, but it blocks the ultraviolet radiation. It blocks the harmful short wavelengths of sunlight. And in combination with the magnetic field, the Earth's atmosphere is important for shielding the surface from particle radiation as well, from this cosmic radiation, these cosmic rays, which are either deflected aside by the magnetic field or absorbed high above our heads by the shielding depth of the atmosphere. So, there's a number of different things, a number of different reasons why the atmosphere is crucial for life on Earth.
Okay, so and without going further to the cosmic radiation, as what your primary research in, what's, for example, the role of UV radiation? That's, for example, is more common on Earth, and we may be approaching to a future will be even more common due to the destruction of the ozone field and everything. Yeah.
Well, luckily, the destruction of the ozone layer has stopped, at least the human-caused destruction of the ozone layer, because we had a big international agreement and things like CFCs were banned for industrial or commercial or domestic uses. So in one sense, the global community's response to the problem of the growing hole in the ozone layer over the poles should give us some optimism that we can also solve another global problem of climate change. Climate change is a much, much bigger problem in terms of the scale of the change we'd have to make to the way the world works to solve. But our response to the hole in the ozone layer should give us confidence that we can work together as an international community to solve global problems that affect all of us. So UV radiation on the whole is destructive. The short wavelengths of light get absorbed by molecules, particularly molecules that have ring-like structures in them, so aromatic structures. So things like DNA or some of the amino acids in proteins very strongly absorb UV and then undergo photo damage. They get damaged. The bonds are broken by the ultraviolet rays. So if you are a life form based on molecules that have ring structures in them, which you almost certainly are, UV is a bad thing, it's a dangerous thing for you. So as a cell, you need to try to protect yourself from that UV, either by using the environment and colonizing inside rocks themselves, which is what things in the Atacama Desert do, what cells, bacteria in the Antarctic dry valleys do, or you can produce natural sunscreens. You can create compounds that absorb the UV before it starts damaging your DNA, and that's another response that biology uses to protect it from the UV.
Okay, that's nice. And regarding combining both of your research fields, which type of organisms would you expect to find on Mars eventually? I mean, if we were able to, or assuming that there is life on Mars, which type of organisms? I mean, some kind of between viruses, bacteria, some more complex organisms?
So for the surface of Mars, we don't, and in fact the solar system as a whole, we don't expect to find extraterrestrial life that is more complicated, more sophisticated than single-celled, hardy life forms, the sort of things like bacteria or archaea on Earth. And that's certainly true for Mars, although there's an outside chance that maybe Europa has been able to evolve multicellular life forms in its ocean beneath the icy face. But that is just speculation. We have no reason to believe that. And so the sort of life you might expect to find on Mars would be extremophilic life. And the Martian surface is very cold, very dry. There's lots of salt, we think, in the soil and the regolith. There's no protection from ultraviolet rays from the sunlight. So Martian life would have to be very desiccation tolerant. It'd have to be very radiation tolerant. It would probably have to be halophilic, so able to tolerate high salt levels. It might need in certain regions to be acidophilic, able to tolerate high acidities, low pHs. So you can look at the environment in different places on Mars today, or what we think that environment would have been like on primordial Mars when it was much warmer and wetter, and therefore understand what life would need to be like to have survived in that situation. And therefore that informs us what sort of biosignatures we'd be looking for.
Okay, and finally, I think, what do you think about the question of if we could or we be Martians, and not because of any alien stuff itself, but like regarding the period of time a few billion years ago, when there was a big exchange of material between Mars and Earth?
So the idea here is that in the early solar system, during the late heavy bombardment, there was the inner planets of the solar system. So Mars, Earth, Mercury, Venus were being pummeled by effectively the rubble left over from the building of the planet. So huge lumps of asteroid were slamming down onto the inner planets, splashing off, throwing off great big plumes of rock from the surfaces of these planets. And so it seems likely that single-celled life, bacterial type life, could survive in lumps of rock, blast off one planet, flung through outer space to land as a shooting star, as a meteorite on another rocky planet in the same solar system. And so the idea of lithopanspermia, it's called, you could in principle transfer life from Earth to Mars or Mars to Earth. This idea that in the early solar system, the inner planets were constantly sneezing in each other's faces, that you could spread life like an infection between neighboring planets. So it's possible that if we do find life on Mars, and it turns out to be very, very similar to us, that Martian life is based on DNA, it's based on proteins, it's based on the same sort of membrane compounds, the sort of fatty lipids that terrestrial life uses. The reason for that might be that it's not independent of us. We found our own long lost cousins. We found descendants on Mars of migrants that traveled there from Earth aboard a meteorite billions of years ago during this late heavy bombardment. But perhaps the opposite is also possible. Perhaps life got started on Mars first and then was transferred to the Earth aboard a meteorite. Perhaps we are already Martians. Perhaps we are immigrants that have come to Earth from Mars from the very earliest period of the solar system. And again, this is an idea, it's very interesting, it's very intriguing, but at the moment it is no more than speculation. We have no reason to believe that life on Earth has come from Mars. We haven't even found that there was life on Mars yet.
Okay, thank you so much. Marco, yeah.
Hi. So yes, as you guessed in the beginning, I was going to ask you something regarding to the Venus stuff, the new thing that appeared. So I'm sharing now through the common Zoom chat the new paper that came out recently, basically arguing that the supposed big discoveries that had been done regarding the possible life in Venus weren't as accurate as they could have been. So I am a person who's not very into science, so I was wondering, is this a normal thing to find these kind of mistakes in papers? Is this a big thing that doesn't usually happen, and how can this be avoided?
You know, this is absolutely normal. You wouldn't want to avoid what is happening at the moment with the Venus phosphine story, because that is simply how science works. That is simply how we progress from one discovery and one building up of new information to another. So to summarize what's happened, there is a team of astronomers, scientists, led by Jane Greaves at the University of Cardiff, who used 2 different radio telescopes so that they were confident that they had detected a chemical called phosphine in the atmosphere of Venus. And this is thought to be exciting because phosphine in Earth's atmosphere is either put there by life or by human industry, by factories. We make it technologically. So it was thought that phosphine would be a good biosignature gas. If we find phosphine in the atmosphere of an Earth-like planet orbiting another star in the galaxy, we could have confidence that there is life on that planet because we don't know how to make phosphine with a geological process, with volcanoes. But we do know that phosphine can be made by life. So this team of scientists checked everything they could think of. They ruled out any possible artifact or error that they might have made. They wrote it up for an academic journal. It underwent peer review. So independent scientists who were not involved in that research checked the sums, checked the conclusions, checked that the discussion in the paper followed on from the raw data, and it was therefore published. And what Jane Greaves and her colleagues in that paper basically said is, look, guys, we think we've just detected phosphine. We can't think of a geological process that could create that phosphine, so maybe it's life. We don't know, but that's a possibility at least. What do you lot think? Is there an error in the data? Is there an error in the interpretation and the analysis? Or can we as a community be confident that there is phosphine in the atmosphere of Venus, and maybe we should go and get a closer look by sending more space probes? And what is happening now is perfectly normal. It's perfectly what the original authors had anticipated and wanted to happen, is other scientists now checking the data, they're making observations of their own, and in this case, they're not corroborating the original discovery, they are failing to find evidence for phosphine. So what will probably happen over the next couple of months is there will be a consensus either one way or the other that yes, there probably is phosphine, or no, on the balance of probabilities, it's not there. But we can only have that conversation, we can only have that discussion as scientists if teams of researchers publish their results so that everyone can see what they've got and then start a conversation, either in the pages of journals or in the halls of conferences or during the world of coronavirus having Zoom calls with each other. That's how scientists discuss things and how things are discovered and how we get confidence in these results.
Okay, interesting. The last question I'm gonna ask today is regarding to new discoveries in the moon, saying that it is very possible, if not sure that there's water on the moon. It can be in different shapes, but there's definitely water. So what are the repercussions in a long term that this might have
Well,
for our society in general?
we've known of water on the moon for a while. We've found signs of water ice in craters up by the poles of the moon, so in permanently shadowed craters, places where the sun doesn't ever shine, and therefore it doesn't get too hot and the ice sublimates away and is lost to space. What is new about this more recent discovery is we found water trapped into the surface rock of the lunar surface itself, and not only near the poles, in more equatorial regions. And the reason this is exciting is if you want to start exploring the moon with humans and establishing lunar bases or colonies on the moon, you can't launch from the Earth everything you need to survive. Trying to launch all the oxygen you would need to breathe for years, or the food you need to eat for years, or the water you would need to drink for years would just be impossible. It costs thousands and thousands and millions of dollars for every kilogram of stuff you launch from Earth to the moon. So what you would need to do to establish a colony on the moon is called in situ resource utilization, ISRU. It's just NASA speak for living off the land. You want to find as much stuff on the moon already there that is useful, so you don't have to pack everything into your suitcase and take it with you when you launch from the Earth. And water, of course, is really useful for keeping astronauts alive on the moon. Not only do you need to drink it and water plants that you would need to grow so you can eat, but if you subject water to electrolysis, you can split it into oxygen, which you breathe, and then hydrogen, which you can use as rocket fuel. So finding water on the moon is really, really important for ISRU and for the chances of establishing a sustainable and self-sustaining colony on the moon in the near future, in the coming years and decades.
Good, good. So I think we're now getting into the brief Q&A we had planned. Is that right, Luke?
Yeah, so if anyone else has any questions they'd like to ask Lewis, then you could type them into the chat, and then if you wanted, you could also ask out loud or something like that. So Miguel, you had one earlier on, I think.
Yes, I wanted to ask him, thank you. I wanted to ask him about his views on planetary protection, because there's been some controversy over it. Some claim that going to Mars would ruin the possibility of finding life there. Others argue that if life was there on Mars, it would be very different from the ones on the Earth, and then it would be impossible basically to misinterpret Earth life or Martian life. So what do you think about that?
Well, we don't know yet what life on Mars would be like because we've not found it. It might be very, very similar to terrestrial life. It might be the case that DNA is one of the best options for storing genetic information and passing on when you reproduce. It might be the case that proteins are the best way of building enzymes. Out of all the different alternatives that are available in organic chemistry, it might be the case that proteins are best. So for various reasons, we might expect Martian life to be very similar to terrestrial life. It might have got there, it might have been transferred between Earth and Mars. We might be related and therefore very similar. Or on the other hand, Martian life might be very, very different. It might be very easy to distinguish terrestrial life from Martian life. But until we look and find it, we don't know. It could go either way. So one of the concerns with sending humans to Mars is this problem of contaminating Mars with terrestrial life in terms of planetary protection and trying to make sure that anything we send to Mars is completely clean, has been sterilized and scrubbed of organic compounds and even the kind of dead bodies of cells and bacteria we want to make sure we don't send to Mars. Because if we do find life on Mars, we want to be confident that it is indigenous Martian life and not basically the dirty fingerprints of some lab tech who touched the probe accidentally before we launched it and we find our own contamination. We want to be certain that it's Martian life that we found on the Martian surface. And while it's relatively easy to sterilize a lump of metal, a robotic space probe or a rover, you can't sterilize a human. You would kill a person by trying to sterilize them of bacterial life in, on, and around their body. So the very first time that humans land on Mars, walk down the ladder, take a photograph of the very first human footprint on Mars, take a picture of the flag that they've just planted in the ground, we will almost certainly immediately begin leaking terrestrial bacterial life onto the Martian surface. And if that happens, we might never be able to tell the difference later on if bacterial life detected on Mars is terrestrial contamination or if it's indigenous Martian life. So there is a good argument to be made, I think, that perhaps we should hold back sending human astronauts to Mars until we've had a fair chance of using clean robots and probes to check if there's indigenous Martian life there or not, before we run a high risk of contaminating by sending humans.
All right, that's great. Thanks. Does anyone else have anything they'd like to ask?
I think someone has just taken a question in the chat. If you want to read it.
Layla, did you want to say it out loud, or shall I say it? Okay. Why did NASA choose the asteroid Bennu of all the asteroids that are in space? And don't you think that astronomy should be taught in school?
Two great questions. Question one, I have no idea. I wasn't involved in the target selection for NASA for the OSIRIS-REx mission. I suspect, though, it had a large amount to do with we need to find an asteroid that is relatively easy to get to. So one that is on a similar orbit to the Earth. And in terms of delta V, which is the critical consideration when you're trying to move spaceships from one part of the solar system to another, we needed to find an asteroid that was energetically economical to reach, as well as one that was of a particular category of asteroid that could answer the questions we wanted to ask. So one that might have had carbonaceous material, organic molecules on it. So I strongly suspect those would have been the reasons, although I don't know. And in terms of why don't we teach astronomy at schools, I think we do. I think there are many schools across the UK that do offer astronomy GCSE, certainly. And even if your school doesn't offer it as a formal option, you will absolutely, definitely be able to find a local astronomical society that you can go along, particularly in the moment in winter when it's night earlier in the day. The astronomy societies will allow you to turn up, maybe charge you a nominal subscription fee, membership fee for the year, but on clear nights, you'll be able to go along and look down a variety of different telescopes at targets like nebulae and galaxies and Jupiter and Saturn and Mars and Venus when they're visible in the night sky. So I'd strongly recommend you just just Google for where your local astronomy society is, or check in Sky at Night magazine and do a bit of digging around yourself.
I just have a quick question about what you were saying about leaving traces of bacteria behind on Mars once humans go. Would the worry be, because obviously I'm not sure, I'm only a first year, so I've only just started studying, so I'm not sure if there would be a lot of bacteria on your body that would be able to survive in those conditions. Would the worry be that the bacteria would survive and harm life, or would it be more that it would affect our findings if we find cell debris from bacteria?
Both. So yes, you are absolutely dripping in bacteria right now. And it depends on how you count it, but there's something like 10 times more bacterial cells in, on, around, throughout your body than there are human cells. You are more bacterial than you are human, simply because bacteria are tiny in comparison to big eukaryotic animal cells. And they live all over your skin. They live in your stomach, in your digestive system, and they are falling off your body all of the time. So if we send humans to Mars, there's a chance that because no spacesuit or a space station or a habitat is ever 100% airtight, some of the air will leak out of the space habitat, the spacesuit, potentially with bacteria in it, and therefore potentially terrestrial bacteria will get onto the surface of Mars. Now, you're right that most bacteria on your body would probably die pretty quickly when exposed to very cold, very dry conditions on Mars, but we don't know how many are going to leak out. We don't want there to be any chance that what we detect on Mars is terrestrial contamination rather than indigenous Martian life. And there are a lot of spore-forming bacteria that can produce spores that we know do survive the conditions in a dormant state on the surface of Mars. With the issue of what are known as special regions on Mars, there are places we think there is water close to the surface, and the places where there might be liquid water transiently for short periods of time, where if there were bacteria there, they could divide and replicate for short periods. So we definitely don't want terrestrial contaminating bacteria to get into those places, because they could then grow and replicate.
Okay, thank you very much.
Yeah, that's great. Thank you. Has anyone else got any questions? Or if not, we've just put a feedback form in the chat, and it would be really appreciated if you could fill it out for us.
If there's no more questions, I could maybe follow up on the planetary protection thing, which I'm quite interested in. So I was wondering, on the one hand, what's the possibility that the life forms on Mars that exist would be indistinguishable from the life on Earth, because they have been living separately billions of years, and also we have studied life forms on Earth for quite a while. So what is the chance that the life found on Mars would be indistinguishable from the life that the astronauts would carry?
It is a great question. I would love to be able to give you an answer to it. We just simply don't know. When you only have one example of something to study, you can't even begin to give probabilities on how confident you are that that is the only option, or relative probabilities for alternatives. Because although on one hand, there is a staggering diversity of life on Earth, from hardy bacteria living in volcanic vents on the seafloor, to oak trees, to eagles, to humans and dolphins, on a molecular level, we're all basically identical. We all use the same fundamental biochemistry and molecular biology to survive. We all use DNA, we all use proteins, we all use lipid membranes on the outside walls of our cells. So the question boils down to, is the only way to build an organism by using DNA to store information and proteins to run as enzymes, or might there be other complicated molecules built out of carbon, built out of organic chemistry, that can also perhaps have a double helix structure, perhaps also store a genetic code and then replicate? And the simple answer is, we don't have the foggiest. Even trying to simulate a couple of water molecules on a computer, on a supercomputer, take an incredible amount of time, let alone trying to model a complicated molecule that could store information. So we can't simulate on a computer what alternatives to DNA there might be. And when scientists have tried to look and done chemical experiments in like real test tubes, you can change one or two things about DNA and see if that molecule still works. You can change the deoxyribose sugar in the DNA and perhaps replace it with glucose sugar, and we find that the DNA falls apart and doesn't work at all. But imagine how many different permutations of things you could change to change every single thing about the DNA molecule to see, does it work, yes or no? And that's not even exploring completely different kinds of molecules that could, in principle, store information. So there's a very long-hand way of saying there's an enormous number of different molecules that you could build using carbon, oxygen, nitrogen, phosphorus, sulfur, and we have no way of knowing in this enormous chemical space which of them are viable molecules for storing and transmitting information. We know of one example, DNA and RNA, which life on Earth uses. So we can't even begin to come up with a probability that life on Mars will be different or similar, because we've got nothing to compare it to. And in a sense, that's the most important reason to look for life on Mars and check, is it identical to us or is it different? Because if we do find life on Mars and we do find that it's different, it's definitely an independent origin of life, and it's not just contamination from Earth billions of years ago, we now know that two next door neighbor planets both independently developed life, and therefore the probability of life starting is probably very high, and therefore the probability of life on other planets across the galaxy is probably also very high. But if we only have one example, if we only have life on Earth, you cannot begin to make up any probability at all, because you have only one example to look at.
And finally, when would you feel confident of humans going to Mars? Because I've also read that in the selection process of all the locations for landing rovers and probes, the best places where life could be found were also sometimes deselected because of the chance that the rover might contaminate the sample. So that might be like the snake that bites itself, because if you cannot send probes to the places where it's most likely to be life, and you don't find life, so when would you be confident to send humans?
Yeah, so you're absolutely right that the principles of planetary protection and trying to be certain that we don't accidentally contaminate Mars with terrestrial bacteria, especially in the sort of environment that life could potentially grow and divide in, that's basically mutually exclusive with astrobiology and looking for life on Mars that could be there. So we try to balance those two considerations against each other as best as possible by doing the best job that we can to sterilize rovers that we send, sending them to interesting places on Mars that might have preserved biosignatures or fossilized Martian bacteria, but not necessarily places where there could be liquid water transiently present today. So we look for past signs of life and not necessarily where life might be able to survive today. But what I suspect might happen is that these international legal frameworks of things like planetary protection, they were signed back in the 60s and 70s when the only people that could send space probes to Mars were superpowers. It was America and NASA. It was the USSR or Roscosmos as their space agency, and nowadays ESA, the European Space Agency. But what we're seeing now with the privatization of space exploration is that single individuals, tech bros like Elon Musk, who've become eye-wateringly wealthy because of the internet, can now, like some kind of James Bond baddie, make their own rockets, launch them to Mars, and potentially start sending probes, if not even human explorers, in the foreseeable future, in the coming decades. So it might be the case that irrespective of what scientists and astrobiologists might urge caution on, irrespective of international law that have been signed by nations like the US, but not by private individuals like Elon Musk, we might find that private space corporations go to Mars anyway, because they can and because they want to. And it might be the case that there's actually nothing we could do legally to prevent it. And we're seeing something very similar at the moment with the network of Starlink satellites that Elon Musk has been launching. Astronomers are getting rightfully very concerned about this network of very bright satellites ruining the view of the night sky. And there's basically nothing anyone can do to stop people launching satellites. There's no law that you can appeal to prevent someone that has a rocket to launch a satellite if they want to.
All right, that's great. Thank you. I think we've overrun a little bit, but thanks very much for coming. It's been really great, really interesting.
No problem at all. Thank you very much. Thanks for the great questions. They're all really interesting. Love to meet you all.
Thank you.
Thank you
Cheers.
so much.
Bye-bye.
Thank you so much.
Bye.
Very much. Goodbye.
Bye.